All right, so get ready for this, listeners. Today we're taking a deep dive into how AI is being trained to analyze medical images,
Uh-huh.
specifically those incredibly detailed ones, whole slide images they call them,
Right.
or WSIs for short. Think of them as like super high-resolution digital scans of biopsy slides,
Yeah.
the ones pathologists use to diagnose cancer.
Yeah, exactly. They're huge, absolutely packed with information.
Oh, yeah, tons of visual information.
A treasure trove, really,
So,
about the tissue, everything, all kinds of stuff about the cells, how they're arranged, and even those really subtle changes that could be like
uh-huh.
signs of disease.
Yeah, so it's like you're trying to find a needle in a haystack,
Yeah, right.
but the needle is microscopic and the haystack is a whole slide.
That's a good way to put it.
So how do you even begin to teach AI to make sense of this stuff?
Well, it's a huge challenge. Training AI for this kind of analysis,
Yeah.
it needs mountains of data. And to get all those images labeled by, expert pathologists, it's a huge bottleneck. It's super expensive and it takes forever.
Right. But that's where this research we're looking at today comes in, right? They've
Yeah.
got a really clever solution.
Yeah.
They're using something called self-supervised learning, or SSO.
Oh, yes. Oh, yeah.
To be honest with you, I'm a little fuzzy on how that actually works. Can you just
Sure.
help me out here?
Think of it this way. Like you're trying to learn a new language,
Okay.
but you don't have a dictionary.
Okay.
So you just start observing how all the words are used in different sentences, right?
Yeah.
Picking up on those relationships between them.
Yeah.
That's kind of what SSL does for AI.
Okay.
It lets those models learn from huge amounts of unlabeled data, which is a game changer in fields like this, where labeled data is hard to come by.
So instead of needing humans to label every single image,
Yeah.
the AI just kind of spots patterns and relationships all on its own.
That's a great way to put it. It's like it's developing a visual language of tissue.
Oh, I like that.
Yeah. And this approach has already shown a lot of promise in histopathology, but here's where things get really interesting.
Okay.
A lot of the existing SSL methods, they rely on tricks like flipping or rotating the image,
Okay.
which helps with, generalization.
Right.
But it doesn't always capture the relationships between different parts of the tissue.
So you're saying that the spatial arrangement of the cells within a tissue sample, that's really important.
Very important.
And some of those existing methods might be missing those cues.
Exactly. That's where this new research comes in.
Okay.
They've developed an SSL method that specifically taps into the natural organization of adjacent patches in the WSI.
Okay, so instead of messing with the images, they're training the AI to see how different parts of the WSI relate to each other.
Right.
Spatially.
It's all about understanding context.
Okay, I'm hooked. Tell
Okay.
me more about how they actually do this.
All right. So it all starts with what they call a pivot patch. They sample this from the WSI. You can think of it like a starting point on a map.
Anchor point.
Then they identify what they call neighbor patches that are, spatially close to that pivot patch.
Okay.
And the key here
Uh-huh.
is that the AI model is trained to produce simile representations
Gotcha.
for both the pivot patch and the neighboring patches.
So if the patches are close together in the WSI, the AI should learn to, represent them in a similar way.
Yes.
It's like building a little map in its mind.
Exactly.
Okay, that's clever. But how do they decide what spatially close even means?
Okay.
Is there a specific distance they use?
They use something called the Chebyshev distance to measure spatial proximity.
Chebyshev distance.
Yes.
I'll be honest, my math skills are a little rusty.
No worries. It's not as complicated as it sounds. Okay, so imagine you're looking at a city grid, right? And each patch is like a city block.
Okay.
The Chebyshev distance is the shortest number of blocks you'd have to travel. To get from one patch to another, you can move horizontally, vertically, or diagonally.
So like finding the most direct route.
Exactly.
Okay.
But why is this the best way to measure distance in this case?
Yeah.
Well, WSIs are created by, scanning the tissue in a really systematic way.
Yeah.
So they naturally have this grid-like structure. So using the Chebyshev distance makes sense because it aligns perfectly with how the image is organized.
Okay, I see the logic there.
Yeah.
So the AI is learning to group similar patches together based on their location in the WSI. But
Right.
what's to stop it from getting too fixated on those specific arrangements that it sees in the data that it was trained on? You know what I mean?
Good point.
Would that make it less accurate when it's trying to analyze new images?
You're right to point that out. That's a risk with any AI model. And these researchers, they tackled this head on.
Okay.
Their method actually uses a combination of two types of pairs to train the AI.
So it's not just spatial context.
Right. They use those contextual pairs that we just talked about. But they also throw in what we call augmentation-based pairs.
So they still do some of that flipping and rotating.
Exactly. This helps the model learn to spot those patterns that are consistent regardless of the transformations.
Gotcha. So it's like teaching it to say, okay,
Right?
even if the image is flipped or rotated,
Uh-huh.
these features, they're still important.
Exactly.
So it's a balancing act
Yeah.
between learning from spatial context
Mm.
and being able to generalize to images that might look a bit different.
I like that. It's like having your cake and eating it too.
And the balance between those two types of pairs, how do they manage that?
Well, it's controlled by what's called a hyperparameter.
Okay,
Think of it like a dial.
gotcha.
They can adjust to fine-tune how much emphasis the model puts on each type of learning.
Makes sense.
Mm.
So they've got this clever method for teaching AI to understand spatial context
Yes.
in WSIs.
Uh-huh.
But the big question,
Yeah.
does it actually work?
That's the million-dollar question, right?
Yeah. Did they see improvement?
So this is where it gets really exciting. They put this method to the test on multiple data sets, and they found that their spatial context-driven method consistently outperformed those standard SSL techniques.
Okay, hold on. You're saying like a 5–10% improvement in accuracy?
We're talking a 5–10% improvement.
When you're dealing with cancer, that could be huge.
It's huge, especially when you consider that.
Catching a tumor early.
Early, yeah.
Wow. Okay. So you mentioned some public data sets.
Yeah, so they tested their method on Chameleon-16,
Okay.
which is a data set of lymph node sections.
Huh.
They use it for detecting metastases in breast cancer
Okay.
and also TCGA-NSCLC.
What's that one?
That one focuses on lung cancer subtypes.
Lung cancer. Okay.
Yeah.
So how did it stack up against those existing approaches on those data sets?
Well, on Chameleon-16, they saw about a 4% improvement in accuracy with their spatial context method
Okay.
compared to the standard SSL techniques.
That's noticeable, a noticeable improvement. What about lung cancer?
On TCGA and SCLC, the results were even more impressive. They hit accuracy gains of over 10% with their method.
10%. Wow, that's a big jump.
Yeah.
It seems like this approach is especially well suited for lung cancer.
Well, it's too early to say for certain without further research,
Yeah.
but the initial results are very promising.
Yeah, for sure.
And in addition to these public data sets,
Uh-huh.
they also use some private data sets of stomach and colorectal WSIs.
Interesting. What kind of results do they see on those?
Well, the improvements on the private data sets were even more dramatic.
Oh, really?
Yeah, on the stomach data set, they saw accuracy gains of up to 8%.
Uh-huh.
And on the colorectal data set, the gains were over 10% with certain SSL methods.
Wow. So consistent improvements across all the data sets they tested.
It seems like it, yeah.
It really seems like this spatial context-driven approach is a major step forward for SSL, for histopathology.
I think so too, yeah.
Okay, so to really understand why this method works so well, they dug into how the distance between those neighboring patches impacted the results.
Right.
So they tweaked that Chebyshev distance
Exactly.
to see how it affected things.
They found that the shorter the distance between the pivot patch and its neighbors, the better the classification accuracy.
right.
Remember that city grid analogy?
Yeah.
The closer those patches are,
Yeah.
the more likely they are to be related in a meaningful way, like part of the same neighborhood.
Okay.
And when they looked at patches that were farther apart, the accuracy tended to drop off.
Yeah, that makes sense.
They think this is because patches that are too far away are more likely to be from different types of tissue, and they don't provide as much useful information for classification.
So by really zeroing in on those spatial relationships between nearby patches,
Yeah.
it allows the AI to build a more accurate picture
Exactly.
and a more nuanced picture.
So this is all incredibly fascinating. It feels like we're getting a peek into the future of AI and histopathology. What does all of this mean for how we train AI models to analyze these medical images? What are the potential implications here? I think this research has some really exciting implications for, how we train these models. It's not just about making them more accurate. It's about giving them a framework for learning from large, unlabeled data sets.
Yeah.
In a way that's more kind of biologically intuitive.
So is making AI more efficient
More efficient.
and cost-effective
Yes,
by reducing our reliance on all that labeled data,
exactly. Yeah.
which is a major win.
Huge win.
But what about the practical applications of this technology?
Sure.
Could this lead to faster, more accurate diagnoses? Could it help pathologists make, more informed treatment decisions.
Absolutely.
There's so much potential here.
Absolutely. It all comes back to that idea of spatial context.
Right.
By teaching AI to see the world more like a pathologist,
Yeah.
focusing on those relationships within the tissue, we're unlocking a whole new level of understanding.
This has been a really incredible deep dive so far. We've covered so much ground.
Yeah.
The challenges of the labeled data.
Yeah.
The power of self-supervised learning. The importance of spatial context.
Right.
We've only just scratched the surface.
There's so much more to explore.
In the next part, we'll take a closer look at some of those specific results from their experiments.
Yes.
And really dig into the implications of this research.
Sounds good.
So stay tuned.
Do-do-do.
Okay, so before the break, we were talking about the pretty impressive results that this new spatial context-driven approach is getting.
Yeah.
You mentioned like a 5–10% increase in accuracy, but I'm curious about those specific numbers from their experiments.
Sure.
Can you break down some of the findings for us?
Absolutely. So let's start with that private stomach WSI dataset. Using a self-supervised learning method called Barlow twins, they achieved an accuracy of 89.3% with that spatial context approach.
Okay, so that's pretty high already.
It is.
What about the results without using their spatial context approach?
Well, with the standard approach, the accuracy was only 82.7%. So just by adding in that spatial context, they boosted the accuracy by nearly 7 percentage points.
Wow, that's a pretty big jump.
Yeah, it's significant.
Did they see similar gains when they used other SSL methods?
Yeah, they did. Similar improvements were observed with methods like BiWAL-L and VikeReg.
Okay.
For instance, with VikeReg, the accuracy jumped from 83.1% to 89.3% on that same stomach dataset.
It really seems like this approach is bringing something new to the table, not just like a small improvement, but something more significant
Yeah, I agree.
across different methods.
Yeah.
What about the other data sets?
Right.
Did they see similar patterns?
The improvements weren't limited to just the stomach dataset.
Okay.
On that private WSI dataset, they actually saw even bigger gains.
Really?
With VikeReg, for example, the accuracy jumped from 80.7% to a remarkable 91.6%.
A 10% increase.
Yeah.
That's incredible, especially when you think about how complex these whole slide images are.
They're very complex.
To see that big of a jump in accuracy is really shows how effective this new method is.
Yeah, it's quite remarkable.
It shows how important it is to teach AI to understand those spatial relationships within the image, just like a human pathologist would do.
Yeah.
So it's not just analyzing these little bits and pieces in isolation. It's about how everything fits together.
Absolutely. Understanding the bigger picture.
That's one of the things that makes this research so exciting, right?
Definitely.
It's not just about making AI, more technically good, but it's about aligning it with how humans actually approach these images.
Yeah, it's a more intuitive approach.
Okay, so beyond accuracy, were there any other interesting takeaways from their research? Did they look into how this spatial context approach affects other aspects of the model's performance?
Yeah, that's a great question. One of the really interesting things they found was that this approach not only improved accuracy, but also seemed to make the model better at generalizing to unseen data.
So it's not just memorizing patterns in the data it was trained on, it's actually learning something more fundamental about the structure of the tissue.
Exactly. It's learning something fundamental.
Wow.
And that ability to generalize, it's really crucial for real-world applications.
Right, because in the real world, the AI is going to encounter all sorts of variations.
Absolutely.
And new cases it's never seen before.
Right, and it needs to be able to handle that.
This all sounds so promising, but I'm also wondering about potential limitations.
Sure.
Are there situations where this spatial context approach might not be as effective?
That's a good point.
Or types of tissue where it might not work as well?
Yeah, it's important to remember that this is still an active area of research.
Yeah.
And there are always limitations to think about. One potential limitation is that the effectiveness of this method could depend on the specific characteristics of the data set.
Okay.
For example, if the tissue samples are highly heterogeneous, meaning they have lots of different cell types and structures all mixed together, it might be harder for the AI to pick up on those spatial relationships.
Yeah, so the more complex and varied the tissue, the more challenging it might be for the AI to find those patterns.
Yeah, that's a good way to put it.
And then another challenge to think about is the computational complexity, right?
Yeah.
I mean, analyzing all those relationships between patches, that's going to take a lot of processing power.
It does, especially when you're dealing with these, huge, high-resolution, whole slide images.
So there are definitely some hurdles to overcome
Yes.
as this technology continues to develop.
But the potential benefits are they're hard to ignore, right?
Oh, for sure.
Faster, more accurate diagnoses.
Less reliance on all that labeled data.
Less reliance on that expensive and time-consuming labeling.
And even potentially new insights into how diseases work.
Yeah, at a cellular level.
This is really exciting stuff.
It is. It's a very exciting time to be working in this field.
So when we zoom out and look at the bigger picture here, this research is part of a broader trend towards what are called foundation models for computational pathology.
Foundation models, yeah. Think of it as a kind of super smart AI
Okay.
that's been trained on tons of data, not just images, but also text, genetic information, all sorts of stuff.
Wow.
And it would have this really deep understanding of biology that could be used for a whole range of tasks in pathology.
So like a general purpose AI expert for all things pathology.
You got it.
That's amazing.
So where does this spatial context research fit into all of this?
Right.
Well, this type of spatial context-driven learning could be a key part of those foundation models.
Okay.
By teaching AI to understand those spatial relationships within tissues, we're giving it a crucial piece of the puzzle
Right.
for developing a more comprehensive understanding of disease.
It's like giving the AI a whole new way of seeing the world, a
Exactly.
way that's much closer to how human experts interpret these images.
Yeah, that's a great way to put it.
This could have implications far beyond just diagnosis.
Oh, absolutely.
Impacting research, drug discovery, even personalized medicine.
Yeah, all sorts of areas.
Okay, so to really dive into their findings here,
Yes.
let's talk about their, what they call ablation studies.
Ablation studies. So think of them like controlled experiments where you remove or disable different parts of a system
Okay.
to see how that affects the overall performance. It's a way to isolate the effects of the different components.
Like taking a machine apart piece by piece to see how it works.
Exactly.
Okay, so what did they learn from these ablation studies?
One of the things they investigated was the impact of that hyperparameter we talked about earlier, the one that controls the balance between contextual pairs and augmentation-based pairs.
Right. The one that determines how much the AI focuses on spatial relationships.
Right.
Versus learning to generalize to all the image variations.
Exactly.
What did they find?
They found that a mixture of both types of pairs generally worked best. If they relied too heavily on one or the other,
Okay.
it didn't work as well.
So it's about finding that sweet spot.
Yeah, finding that balance.
It sounds like a delicate balancing act.
It is. It highlights how important it is to carefully fine-tune these hyperparameters
Yeah.
to get the best performance out of these models.
Every little detail matters.
Every little detail matters.
When you're dealing with something as complex as medical image analysis,
Right.
what other insights did these ablation studies reveal?
Well, they also looked into that Chebyshev distance
Okay.
and how they defined those neighboring patches.
Yeah, what'd they find?
They discovered that using shorter distances between patches consistently led to better classification accuracy.
Makes sense.
The closer those patches are, the more likely they are to be related in a meaningful way and provide useful context.
Just like we were saying before, the farther apart those patches are, the less likely they are to be, telling us the same story.
Exactly. And when they included patches that were too far apart, they saw a drop in accuracy, which suggests that those patches were more likely to be from different tissue types.
Right.
And they were actually kind of muddying the waters.
So it's all about finding that sweet spot, right?
Yeah, not too close.
Not too far.
Not too far, just...
Just the right distance to get those spatial relationships.
Exactly.
This spatial context-driven approach, it feels like it could revolutionize the way we use AI in histopathology.
Yeah, it's very promising.
It's a really great example of how combining those biological insights
Yeah,
with advanced AI techniques
absolutely.
can lead to some really exciting breakthroughs.
Yeah, it makes you wonder
It does.
what other hidden patterns AI might be able to find.
Right, if it can understand spatial context this well,
Uh-huh.
what other mysteries could it help us solve?
Yeah, it feels like we're in the verge of a whole new era of discovery in medicine.
It really does. Okay, well, we've covered a lot of ground in this deep dive.
Yeah.
We've explored the challenges of working with labeled data. We've seen how powerful self-supervised learning can be.
Yeah.
We've seen this remarkable impact of this spatial context-driven approach. But there's still more to come.
There's more. In the final part, we'll delve into the broader implications of this research and talk about what the future might hold for AI in histopathology.
Okay.
And we'll leave you with some thought-provoking questions
Sounds good.
to ponder, so be sure to stick around.
Okay, so we've spent this whole deep dive exploring how AI is learning to see like a pathologist, you know, breaking down those really detailed whole slide images, the
Right.
ones they use to diagnose cancer. It's been pretty amazing.
It really has, yeah. We've seen how this new approach to self-supervised learning,
Right.
with its focus on spatial context, it's really producing some impressive results.
Yeah, and not just improving the accuracy, but potentially changing how we diagnose and treat these diseases.
Absolutely.
What's blown me away is that it's not just about making AI more, technically better.
Yeah,
It's about teaching it to think more like a human expert would,
exactly.
to understand those relationships between different parts of the tissue,
Right, just like a pathologist.
like they're looking at a slide under a microscope.
Yeah, it's about giving AI a deeper, more intuitive grasp of the biology, which could lead to more reliable and insightful diagnoses in the future.
So this all leads to a really big question.
Yeah.
What does this mean for the future of pathology? How might this actually change how we diagnose and treat these diseases in the years to come?
Well, I think one of the most immediate applications is going to be in automating certain parts of the pathology workflow.
Okay.
Imagine AI algorithms that can pre-screen these slides, highlighting the suspicious areas
Yeah,
for the pathologist to then review.
that'll be huge.
It would free up pathologists to
Yeah.
focus on the more complex cases,
Yeah.
and it could really speed up the entire diagnostic process.
And it could even help to reduce, diagnostic errors
Yeah, absolutely, because
because these AI algorithms are so consistent.
AI algorithms are incredibly consistent and thorough.
They don't get tired
They don't get tired.
or distracted.
No distraction.
And they can analyze so much data, way more than a human ever could.
Exactly, yeah. It's like having an AI assistant working alongside the pathologist, providing a second set of eyes, helping to catch those subtle details that might be missed.
Yeah, I like that, an AI assistant.
Mm.
And it's important to remember that this technology, it's not about replacing pathologists, right?
No, definitely not.
It's about giving them better tools.
Augmenting their abilities,
Yeah.
helping them make better decisions,
Right.
and ultimately provide better care for their patients.
So it's all about that collaboration between humans and AI,
Yes,
bringing together the best of both worlds
the best of both worlds.
to really revolutionize the field.
It's a very exciting time to be working in this field, that's for sure.
It really is. But with all this excitement, we also need to think about the bigger picture.
Yeah,
There are some ethical considerations,
absolutely.
societal implications as this technology keeps getting better.
You're absolutely right. As with any powerful technology,
Yeah.
we need to be mindful of the potential downsides. How do we ensure that these AI algorithms are fair and unbiased in their analysis? How do we protect patient privacy? Right. All those things, data security. These are crucial questions.
Yeah. And it feels like we're kind of stepping into uncharted territory,
We are, yeah.
and we need to be cautious.
Cautious, but enthusiastic.
Yeah. And make sure these advancements benefit everyone.
Absolutely. It's a balancing act, but it's worth it.
So listeners, this brings us to the end of our deep dive today. We've explored this amazing world of AI and histopathology, and
Yes.
it's been quite a journey.
It has been, and we hope you learned something new and exciting along the way.
Remember, the worlds of science and technology are constantly evolving. There's always something new to learn.
Absolutely.
So keep asking questions, keep learning.
And keep pushing the boundaries of what's possible.
And, as always, thanks for listening.